viewers, welcome back to KIC'S tutorials. In our last class, we talked about some genetic vocabulary like genome, genotype, P1 generation, F1 generation, and others. And I told you that in this class we'll be talking about the cell. This class is basically an introduction of the cell. We'll talk about many other things concerning the cell when we are discussing cell biology in subsequent videos. So, cell. And today we'll be discussing the cell under different headings. First, definition, a brief history of the cell theory, the types of cell we have, and I'll be comparing the types of cell we have. So, starting with the definition. The cell is said to be the basic or fundamental unit of structure and function in living organisms. What this means is that in terms of structure, the cell forms the basis of the structure of an organism. How it does this is that cells combine to form tissues, tissues combine to form organs, and organs with similar function work together to form an organ system, and organ systems in turn work harmoniously to perform different functions that ensure the survival of that organism. That's in the case of multicellular organisms. Organisms with different or many cells forming that organism. And since I've mentioned multicellular organism, we also have unicellular organisms where one cell makes up the bulk of the whole organism. Okay, that brings us to a brief history of cells. I told you in the last class that you will get to know about how we got to know the cell. The cell was first discovered and named by an English scientist named Robert Hooke. In the year 1665. He discovered the cell when he examined thin slices of bottle cork under the microscope. He saw that there were little compartments in that thin slice of cork. So, and he likened these compartments to the rooms inhabited by monks in the monastery. So he brought the term cell, derived from the Latin word cella, meaning small room. Okay, I'm sure we must have heard of cell theory, maybe from someone or from some friends. The cell theory was derived from various observations by different scientists during their studies of different organisms or phenomena. So, the two main scientists that formally articulated the cell theory are two German scientists, Theodor Schwann and Matthias Schleiden. One a botanist and the other one a zoologist. They made some observations while studying their different organisms and discovered that they had some common observations among these organisms that they studied, which brought them to propose the cell theory. The cell theory has three tenets. And the first is that all organisms, all known organisms, or living organisms, are made up of cells. From bacteria to amoeba to humans, we are all made up of cells. Then the next is that the cell is the basic unit of structure and function in living organisms. Just this definition I gave you about the cell, this is where it was derived from. And then the last tenet says that cells arise from pre-existing cells. That is to say that every cell has an ancestor, and they arise from pre-existing cells through the process of cell division, where one cell splits into two to give two daughter cells, and in the case of unicellular organisms, two daughter organisms. Okay, now I'm sure there's a question running on your mind, which is it that there's only one type of cell in the world? Actually, no. There are various types of cells in the world with different shapes and sizes, and though almost the same chemical composition. But broadly or basically, cells are classified into two categories. We have the prokaryotic cells and the eukaryotic cells. Okay. The prokaryotic cells are cells that have a nucleus that is not bound in a membrane, and therefore it is called a nucleoid. It is called a nucleoid. That is the genetic material of such cells is contained in a region that is not separated by a membrane from the rest of the cell. Okay, sorry. Then, organisms with this kind of cells are called prokaryotes. And examples are bacteria like E. coli. Eukaryotic cells. Eukaryotic cells are cells whose nuclei or nucleus is the plural for nucleus, whose nucleus is bound in a membrane. That is, it is separated, its contents are separated from the rest of the cell, which is called the cytoplasm. And such cells, besides the nucleus, have other units, or what we call organelles, that are membrane bound also. And organisms with such cells are called eukaryotes. Okay, that's it for the types of cells. Now we are going to be comparing these two types of cells to see the similarities and the differences between them, which gave the reason for the for putting them in different categories. Okay, I'll be comparing them based on several characteristics which one might possess and the other might not. And the first is cell size. First, cell size. Prokaryotic cells are usually very small, though most cells can only be viewed with the help of the microscope. You can't see them with your naked eyes. But these type of cells are really small, very small, usually 1 to 10 micrometers. They are really tiny, where you would need a special kind of microscope to view them, like the electron microscope. So Eukaryotic cells are much larger, though you still need a microscope to see them. They are much larger, ranging from 10 to 100 micrometers. Okay, next is their genetic system. Okay, prokaryotic cells have DNA in association with some non-histone proteins, actually many different types of proteins that help contract the DNA or condense it to form one single circular protein. Chromosome, please. One single circular chromosome. DNA plus non-histone proteins. We'll get to find out about histones and non-histones when we talk about the cell proper, the genetic material. Okay, also this chromosome is found in a region called the nucleoid. Like I told you before, that's the region that is not bound by a membrane. And then in eukaryotes, the DNA is in complex with histone proteins. And non-histone proteins. Then, as it combines with these histones, it condenses to form different or many chromosomes. And here, the chromosomes are linear, as opposed to circular chromosomes in prokaryotes. So we have several chromosomes. In humans, we have 23 pairs and in total 46 chromosomes in every cell. Okay, and this DNA in complex with the proteins are found in a region of the cell called the nucleus, and it is separated, like I said before, it is separated from other components of the cell. Now the third characteristic, cell division. Prokaryotic cells divide basically by binary fission. And it's a form of asexual reproduction, that is to say that different gametes do not fuse in this kind of reproduction. The cell just splits into two daughter cells. Also, some prokaryotes engage in budding. The cell division in them is in budding, like in Hydra. We have a little bud growing from the parent organism until it's mature enough and then breaks off to be an independent organism on its own. And then that would be all for prokaryotes. There is no mitosis here. But in eukaryotes, mitosis takes place. The method of cell division, or the mechanism of cell division, is mitosis, and it happens in the body cells, somatic cells. Those are the cells that are not involved in reproduction. But in germ cells, that's the reproductive cells, meiosis is what takes place there. In mitosis, two daughter cells are produced from one parent cell. But in meiosis, 4 daughter cells are produced from one parent cell. For instance, during the formation of sperm cells. Alright. Next characteristic is nutrition. Nutrition in prokaryotes is by absorption photosynthesis. Okay, by absorption, most prokaryotes feed on organic molecules like sugars, amino acids, hydrocarbons, methane gas, and so on. And such organisms are called organotrophs. They absorb them into the cell. And then the other the other prokaryotes engage in a type of absorption where they absorb inorganic nutrients from the environment. And such prokaryotes are called lithotrophs. So they feed on inorganic nutrients like carbon from carbon dioxide. They feed on sulfur, some on iron, and so on. So that's for prokaryotic cells. For eukaryotic cells, the modes of nutrition are absorption, ingestion, and photosynthesis. Sorry, I didn't explain that of prokaryotes, photosynthesis. Some prokaryotes are also photosynthetic, like the blue-green algae, which is cyanobacteria. They feed mostly by harvesting sunlight energy and reacting it with some other molecules to produce their food. So eukaryotes feed through absorption. Some feed through ingestion, where the organism takes in food, like in humans, through the mouth, where the saliva and the teeth and everything works on it, and then it goes down to the stomach for further digestion. Eukaryotes also undergo photosynthesis, where they just like the prokaryotes harvest sunlight and then use it for production of food. Okay, that's it for nutrition. The next is energy metabolism. Energy metabolism. Prokaryotes metabolize or produce energy through oxidative enzymes on its cell membrane. Here there is no mitochondrion or mitochondria present in the cell. So those oxidative enzymes do not have where to stay in the cell. They have to be on the cell membrane. But in eukaryotes, we have the mitochondria, which is usually referred to as the powerhouse of the cell, where energy in the form of ATP is produced, and the oxidative enzymes are found enclosed or packaged in the mitochondria. Alright, next, I'll have to just clean this. Next, we have intracellular movement. Intracellular movement. In prokaryotes, there is no intracellular movement. What I mean by intracellular movement is when sometimes maybe the cytoplasm moves within the cell, or the cell membrane engulfs substances from its environment. We can only find that in eukaryotes, and eukaryotes engage in different intracellular movements. For instance, we have cytoplasmic streaming, which we call cytosis as the other name for it. What this means is, in the cell, the cytoplasm moves due to the action of some microfilaments, or like in some cells where the vacuole moves, maybe to store water or regulate the temperature, or most other functions that vacuoles carry out. We'll find out more about that when we are talking about cell organelles. So that's what happens during cytoplasmic streaming. And another form of intracellular movement is endocytosis. Endocytosis is when the cell membrane covers or engulfs, so to say, substances, liquid or solid. And when it is solid particles or microorganisms, we have phagocytosis, where the cell kind of eats up another cell, microorganism or substance. An example of phagocytosis is in the human immune system, where some white blood cells like neutrophils and macrophages engulf microorganisms that are pathogenic to the individual. Then also you can observe phagocytosis in amoeba, where it uses that to feed on some other organisms or particles. Okay, after phagocytosis, we have pinocytosis. This is another form of endocytosis, but here it is liquid substances that are taken in by the cell. For instance, in the cells lining the small intestine of man, there are some cells that take in fat droplets in order to absorb them. So that is the process, pinocytosis, that helps it carry out that function. So next, I have receptor mediated endocytosis. Here, special or specific receptors on the cell membrane or on the surface of the cell trap or bind to specific substances and transport them through the cell membrane into the cell. Usually these receptors are transmembrane proteins. We'll get to know more about these membrane proteins when we are talking about cell organelles and the cell wall in particular. Okay, sorry, the cell membrane. That's it for intracellular movement. Next is flagella. This is the organ that aids for movement in some prokaryotic cells and some eukaryotic cells. Now, in prokaryotic cells, okay, let me start with eukaryotic cells. In eukaryotic cells, the flagellum is composed of two single microtubules. I will explain microtubules in subsequent videos. Two single microtubules are surrounded in a circular manner by 9 pairs of microtubules. That is 2 surrounded by 1, 2, 3, 4, 5, 6, 7, 8, 9. This is called 9 plus 2 microtubule structure. Now, in prokaryotic flagella, you don't find this structure. The flagellum is just the helically shaped structure, not like this. So this is a major difference between them. Now, the next characteristic is the cell wall. The cell wall in eukaryotic cells is composed of disaccharide polymers. There's some sugars in association with some proteins. But in prokaryotes, the cell wall does not have this composition. And mind you, eukaryotic cells, it is not all eukaryotic cells that have the cell wall. The cell wall can only be found in cells like plant cells. But in prokaryotic cells, almost all of them have the cell wall. Okay, there are so many other differences between these two types of cells, but I won't be discussing them here. And then you might want to ask, are these cells so different that there is nothing that can be used to relate them? No, they are not different in all ways. For instance, they have similarities, like they both have DNA, that's the deoxyribonucleic acid, which is their genetic material, although some have maybe RNA as genetic material, but mostly the DNA is their genetic material. And then they both have the same genetic code. That is to say, what the sequence of DNA that codes for a particular amino acid or protein in a eukaryotic cell is the same sequence of DNA that will code for that same amino acid or protein in a prokaryotic cell. Then next, they both synthesize proteins, though with almost the same mechanism, but in different ways or in different sites of the cell. They both produce proteins. Then molecules like ATP perform similar roles in both of them. There are so many other similarities, you can find them out for yourself. That brings us to the end of today's class. In the next class, we'll be talking about genetics proper. I'll be entering genetics, Mendelian genetics, principles of dominance, and all the rest. So please don't forget to give this video a thumbs up, share with your friends, hit the notification button so that whenever we upload a new video, you will be updated. And if you have not, please subscribe to the KIC'S tutorial channel to help us make more videos like this. Thanks for watching. See you next time.